going to be talking about excitation-contraction coupling. Excitation-contraction coupling is basically a complex sequence of events that is going to trigger muscle fiber contraction. So it all begins with the excitation of a motor neuron, and it's going to end with that muscle fiber contraction. So to start this process, we begin with action potential. Well, where does it come from? Starts in your brain. Action potential comes from your own noggin. So the action potential is going to start in the brain, and then it's going to go on to the alpha motor neurons. And then from there, it's going to arrive over here. So if we look over at this image, it's going to come to these little guys right here, which we like to call the dendrites, or the dendrite receptors that are on the neuron cell body. So these guys are like little gloves. That's what you can think of them as. They like to grab. They like to grab. That's what a dendrite is. So then after it goes to the dendrites, it's going to go on down to this guy right here. And this guy is the axon terminal. Same thing as over here. Just two different versions, two different ways to look at it, but this is the axon terminal. So it's going to come down to the axon terminal, and we're going to have ACH, or acetylcholine, whichever you like to call it. And that's these little red dots right here. So these little red dots are going to travel through that axon terminal, and they are going to be released. And they have to cross the synapses and bind with the neurons on the plasmalemma. So a different kind of way to look about it, to help you understand really, is that these little guys, ACHs, the red guys, are the passengers, okay? They're people, and they're carried by these things that we call synaptic vesicles. So these guys right here, these little black circles, are synaptic vesicles, and they're going to be our cars. So the Ubers, or the cars, are going to take us down, take these passengers, or the ACH, down, and they're going to bring them to this destination right here. They're going to bring them there, they're going to drop them off, and now the ACH is on its own. Those passengers are on its own. And they have to cross the synapses and bind. There's a little space in between right here. They're not actually like touching. And we call this the synaptic cleft, and that's just right on in here. That's going to be the cleft and the synapses that they cross and bind to. So, the action potential is going to continue on once it has crossed this little gap that we have here. It's going to go on down to the plasmalemma, and it's going to go on down to the T-tubules. So over here, if you can see, we have the T-tubules. So those little guys are going to come all the way down the T-tubule. So the action potential gets into this interior part of the cell, and this is ultimately going to trigger the next event in the sequence. And that brings us to this blue guy over here. This is the DHP receptor. So this receptor is like a little gate, I'd say, and it's going to control the ability of the calcium ions to leave the SR, or the sarcoplasmic reticulum, and get out of there. That's what that gate is. It's opening that so those calcium ions can go through. These blue guys right here are the calcium ions. But we don't want these calcium ions to just rush out the gate. So we have another receptor, and that's going to be the RYR receptor, that's going to control the flow of these guys right here, of this calcium ion. And when that happens, when this calcium is released from the SR, they start going away. Say goodbye. Well, how do you get them back? Well, that's the job of the circa. The circa, which is SERCA, is going to replenish this SR with its calcium ions. It's going to bring them back. And now you have more little blue dots. So now we're going to move on to this thing we like to call tropomyosin. And tropomyosin covers the myosin binding sites. And it's not going to allow these myosin heads to bind. It's not happening. It's not going to let it happen. But then eventually we're going to get the troponin right here, these little circles, and the calcium ions are going to bind on this actin, which is this strip right here, that's actin. It's going to bind on those actin molecules, and it's going to allow the myosin heads, which are these, they look like light bulbs almost. It's going to allow those myosin heads to bind on the sites of the actin molecules. But one important thing that we need for these contractions to happen is ATP. ATP makes it possible for those contractions to happen. And so then these little blue dots come into play again. Those calcium ions are going to enable the actin-myosin contraction. They're going to allow the muscle fibers to contract. And it ultimately ends with the neural cells saying, hey, stop. They're gonna seize it, and that's gonna happen at the NMJ, or the neuromuscular junction. So this right here is just a slight look into the excitation-contraction coupling and what it's gonna look like in that cell and in your body. Thanks.